Hello, everyone, and welcome to the EarthSci show. This is the second episode, and I'm honored to have Dr. Zerina Johanson, who is a researcher from the Natural History Museums in London, Earth Science Department, and she specializes in early vertebrate evolution. And we'll be talking about that and how older species and specimens can determine future theories. And you'll get to find out a bit more about what Dr. Johanson specializes in a few minutes or over the course of this episode. So, Dr. Johanson, thank you for coming. Thank you for your time. How are you doing today?
I'm very well. Thank you for the invitation.
No problem. All right, so let's jump right in. So,
Okay.
the first question I have for you is, so by researching the development of the vertebrate skeleton, which of course you're an expert in, does this provide any key insights into how vertebrates such as ourselves may evolve further in the future, especially given the current climate and how different species are becoming extinct today?
Well, I was thinking about this question, and I thought I would like to concentrate on a group that I'm quite familiar with, which are the sharks and the rays. So the chondrichthyans is a broad group which includes the sharks and the rays, skates, and an unusual group known as the chimera. And what I find so interesting about the chondrichthyans is that their skeleton is made up primarily of cartilage, compared to ourselves or to bony fish, where of course our skeleton is made up of bone. But the chondrichthyans do mineralize their cartilaginous skeleton. So in their backbone, for example, in their vertebral column, they have the central part of the vertebral column is formed by an element known as the centrum. And the centrum is mineralized throughout. But the other parts of the skeleton have this very fine or very thin layer of mineralization on the top of the cartilage, which is known as tesserae. And one project that I'm wanting to develop with my colleagues is to look at how the mineralization patterns in the chondrichthyans have changed through the fossil record to the current day. So of course, the thing that preserves best in chondrichthyans are the teeth and the scales or the denticles, but you do find examples of the skeleton preserved in the fossil record as well. So our idea is to look at the mineralization in living chondrichthyans, the sharks, skates, and rays primarily, and try to correlate that with very specific environments to see if they're, so if you have like a warmer environment, do you have a certain type of mineralization, or a colder environment, again, a certain type of mineralization. So for example, in the deep sea, we know that in general, chondrichthyans have a less mineralized skeleton. So we could take the what we see in chondrichthyans in the present and then try to apply that to what we see in the fossil record. So chondrichthyans have a very deep fossil record. They go, in fact, all the way back to the Ordovician. So we have a long history to play with. And so there hasn't been much work until very recently about how the environment affects mineralization of the skeleton. And so I was just reading some papers recently which have looked at things like temperature, but also acidification. So ocean acidification is a problem today that we face with the environment and environmental challenges. And the paper that I was reading, it was quite interesting because it said that they did a test where they controlled the levels of acidification in the water that these, I think they were skates or rays, were being kept in. And they found that with increasing acidification, they found that there was more mineralization in the chondrichthyan skeleton.
Okay.
So there's some reaction to the acidification that causes, they believe, or they hypothesized, that causes the skate to release phosphate into its bloodstream, which becomes deposited as mineralization in the skeleton. So that could be a real impact. It could be good, it could be bad. So for example, mineralizing a skeleton strengthens the skeleton, which is good, but it also makes the skeleton heavier. So that could be a real disadvantage for an animal that swims if it finds itself having to expend more energy to swim. So I think in that sense, those kind of observations on the skeleton and chondrichthyans in the modern record can really tell us about how an animal might react if environmental conditions change. So, and then we can apply that back into the fossil record, as I was saying, to see if there are any changes that might be linked to major climatic events or major extinction events.
Yeah.
And so we can, by moving through the fossil record to the present time, I think we can start to build up a real record to see how the skeleton of these already quite endangered animals can be affected by things like climate change and ocean acidification.
Yeah, that's very interesting because, and that's quite current, given the climate that we're living in at the moment, because ocean
Absolutely.
acidification is very big and prominent.
Well, in that sense, and I think also with respect to just climate warming, there's been more work done on bony fish. So there's a great opportunity, I think, to be looking more closely at the chondrichthyans or the cartilaginous fish as a group that is quite heavily impacted by extinction today.
Yeah, I mean, considering they've been around for so long, as you said, since the Ordovician,
Yeah.
I don't think enough research is being done. Everyone's quite interested in the bony fish, because they evolved from placoderms and they're our ancestors. But I think the real magic lies in the cartilaginous chondrichthyans that we see today.
Well, one thing is that, of course, it's very easy to keep bony fish in a lab. Zebrafish is an extremely common lab animal. And it's really only in the last decade or so that the little skate, Leucoraja, has been used as a model organism in the lab. And I believe this study that I was telling you about was on the little skate. So as work develops to be looking at chondrichthyans as these lab animals, I think we'll be able to be doing more this type of research across a broader range of chondrichthyans as well to see how different groups respond.
Yeah, that sounds quite good, actually. So especially relating that to what's going on in the planet right now and advancements in technology and being able to hold a wider range of specimens than before. I think that would definitely help us to understand more about chondrichthyans, and them being present today and the extinct chondrichthyans, that will help us to understand the hopefully evolutionary trajectory of some of the extant ones and also predicting the future in some sense.
We were also interested in looking at how adaptable chondrichthyans were. So how quickly or are they even able to modify the mineralization in their skeletons? So the study I told you about suggests that they can, but are they able to, as environments warm up, are they able to change the mineralization within their skeleton, or do they have to seek colder climates because they aren't able to adapt their mineralization? So the skeleton, of course, is so important for locomotion, for example, and just being able to adapt in that way, we think is very important, but we just don't know if sharks can do it.
Yeah. I mean, I guess that process of evolving to those specific climate conditions would just take millions of years. I mean, it doesn't just happen overnight. So, I mean, that would take time. But I think considering the fact, as you said, because chondrichthyans have been around for so long, I think they'd be able to adapt to it fairly quickly because they've seen quite a lot over the period of time.
True enough.
Yeah. All right. Thank you for that. So the next question is, what do you think are the benefits of doing what you do? So researching and finding more about how chondrichthyan dentition formed. And for the audience members who don't know what that means, it's how the teeth of cartilaginous fish formed.
Well, we should all take a lesson out of the chondrichthyan playbook, because sharks and rays, excuse me, and skates can produce teeth throughout their lives. So I think most people would be familiar with sharks' teeth and seeing Jaws and nature programs,
Yeah.
and how important the teeth are for shark feeding. But what happens during development or during the growth of the animal is that if you can picture the jaw of a shark and go to the bottom of the jaw, that's where the new teeth are forming. So they form in tissues down at the base of the jaw, and it's almost like a conveyor belt or an escalator. So the jaw of the shark is covered in teeth from the bottom of the jaw up to the top. And so perhaps you can picture teeth developing at the base of the jaw and developing cusps and mineralizing as it moves up towards the top of the jaw. where it becomes functional in feeding until at a certain time, whatever is holding the tooth to the jaw is lost and the teeth fall out. And because of this, the sharks always have a functional tooth, or more importantly, a set of functional teeth along their jaw, which is very important for their feeding. Rays and skates do this as well, even though people may not be quite as familiar with them, they also produce teeth at the base of their jaws, which move forward to be functional and then fall off. Now, my colleague at the University of Florida, Gareth Fraser, we've been working together for several years now, and what he's interested in is finding the genes that are responsible not only for tooth development, but tooth regeneration, as he calls it. So producing teeth at the base of the jaw is also, in a sense, tooth regeneration, because teeth are lost, they're regenerated at the base of the jaw. So his work has produced several genes that are involved together, not only to develop the tooth, but to very carefully position the tooth along the jaw. So if you think of, just picture a shark jaw in your head and that battery of teeth, it's very precisely positioned. Every tooth on that jaw has a very specific position, so that when the teeth do move up to the top of the jaw, you have the series of teeth along the top of the jaw, then they're lost and the next series comes up. So that's really important, not only the development, but the positioning as well. But Gareth's work is also looking at the stem cells that are involved in tooth development. Now, stem cells are quite important because they're sort of multipotent cells that never die. And so they're available continually to produce tooth after tooth after tooth. Now, what Gareth would like to do, and I think this is such a great idea, is to take what he's learned from sharks and how sharks produce teeth in such great numbers over and over and over again, and see if he can, in the future sometime, apply that to humans. Now, wouldn't that be great if we could find some way to genetically manipulate our teeth to allow us to produce more than the adult number of teeth that we have? So you can see that could, I mean, we don't want to suddenly have a jaw full of shark's teeth, but wouldn't that be great if we could, lose a tooth, but then be able to quickly produce another tooth throughout our lives? We wouldn't have to have, we wouldn't have cavities, we wouldn't have this and that, and all these dental problems that we have already. So I think that's one of the really exciting things that is happening at the moment in the study of shark teeth that has, I think, will have a really direct and important impact on us, on humans.
That sounds quite interesting, actually. I didn't expect that when I asked the question, to be honest.
Oh, okay.
I mean, you learn something new every day. But when you talk about that being applied to humans, I immediately think of the further benefits of that as well. So say if you break a bone, or if you are involved in like a really bad accident, then the potential for using such genes in such scenarios is just infinite, in my opinion, because the medical potential is just great.
Sure, it's just being able to have your tissues repair themselves whenever you want, something that you can control, perhaps speed up, just make more efficient. Yeah, but I think with teeth, it's kind of something that, well, certainly at my age, I would be very much interested in being able to produce more teeth. And it's just, I think it's really exciting that we can take what we learned, because, I mean, chondrichthyans are vertebrates. Vertebrates are animals with backbones. We are also vertebrates. So the genes that are influencing tooth development in chondrichthyans, we will have as well. So just being able to manipulate genes like that, I think is really exciting, and very good on Gareth to be able to do that work on chondrichthyans that might have this amazing application for the future.
That sounds great. Yeah, I think I'll definitely be looking more into your colleagues' work, and I urge the audience to do so as well, because you never know what you learn. And I think the main, point of this episode is to really highlight to the audience who are or aren't in the paleontology community just how intricate it is, and how relevant it is to the wider life that we live. And it's not just, a dead subject, as many people like to consider it as. So, yeah, that's really good to hear, actually, just the potential of that, of a paper.
Mhm.
Yeah. Okay, thank you for that. Um, so so many people know about the Devonian extinction. So you have the Hagenberg event, and so the Kelwasser. I discussed
Yeah.
this with Professor Benton last time in the last episode. But so what impact do you think those events would have had on anather stone radiation? And again, for our audience doesn't know what that who don't know what that means, anather stone is a jawed animal. Nathos is a jaw in Greek, and radiation is them spreading out across the globe. So, Dr. Johanson, what do you think would be the impact on that?
There was a very good study done, I think it was in 2010 by Lauren Stalin and Mike Coates,
Yeah.
when she was a PhD student at the University of Chicago in Mike's lab. And they looked at that very subject. So what they did was they did a broad database survey of all taxa across the vertebrates, when they lived and when they died. And what they found was, with respect to these two extinctions that you mentioned, I think it's the Kelwasser that's in the Devonian, between the Franian and Flaminian,
Yeah.
and the Hagenberg is at the end of the Devonian. What they found was quite interesting, because the Kelwasser, I believe, is supposed to be one of the big five extinction events. So one of the major extinction events that caused a lot of extinction amongst animals. But
Yeah.
it looks like it's the Hagenberg at the end of the Devonian that really impacted gnathostomes. So what they found was that placoderms go extinct. Sarcopterygian fish are very badly affected. So sarcopterygian fish are what we also know as lobe-finned fish. They include things today that, like the lungfish that lives in Queensland, for example, and the coelacanth, which
Yes.
probably a lot of your listeners have heard of. One of the descendants of sarcopterygian fish are tetrapods, so four-legged animals like ourselves. So like Neil Shubin says, you are a fish because you have descended from sarcopterygian fishes.
Yeah.
Anyway, the fish themselves, but not the tetrapods, became very reduced in numbers after the Hagenberg, as did the acanthodians, which are often called the spiny sharks. They also continue on beyond the Devonian Carboniferous boundary, but their numbers are very reduced. So placoderms extinct, sarcopterygian fishes, and acanthodians greatly reduced. But the groups that do flourish after the Hagenberg are the bony fish, the actinopterygians, and also the chondrichthyans. So some groups have done very well, but other groups have done very poorly. And again, one of the things that Stalin and Coates found was it wasn't the big five that caused what they refer to as this bottleneck, but it was the subsequent extinction at the end of the Devonian into the Carboniferous period that caused the placoderms, one of my favorite fish groups, to go extinct.
Okay, that's more interesting. So, so what specifically would you say about the Hagenberg, considering it happened later? What specifically about it would you say caused a dramatic impact and change of marine biodiversity?
Well, I'm not sure myself. Maybe Mike Benton had something more to say about the causes of the Hagenberg. Was it the Kelwasser that has something to do with? I don't remember now.
Was it Oceanoxia, I think?
Was that the Kelwasser? Yeah.
Yeah, no, that was the Kelwasser. Yeah, yeah.
Yeah. So you would think that that probably would have had this more deleterious effect on the gnathostomes, but it doesn't appear to be the case. It appears that this Hagenberg extinction is more problematic. But one thing Stalin and Coates do know is that maybe there isn't enough sampling of the gnathostomes at the Franian Flaminian boundary for the Kelwasser. And so, maybe future work will show that it was also quite a bad extinction event. But for what we know now, it seems to be the Devonian Carboniferous is the real time when some major groups of fishes were badly affected.
Yeah, that's interesting. I mean, many people say that the Permian Triassic extinction is the worst because it
Yeah.
wiped out like 96% of all life on Earth. But
Yeah, indeed.
I think,
Yeah,
in my opinion, I think the Devonian is the worst because it killed off so many fish species.
yeah, yeah.
So many fish.
Tetrapods do well. It's just that the Starcopterygian fish are badly affected.
Yeah, exactly. But then again, you still have some descendants today, as you said, like lungfish in the
Yeah.
sea.
So uh, I don't know if many of your viewers know much about placoderms, but they were an extremely successful group of jawed vertebrates. Um, so perhaps that group going extinct presented all these new environmental niches and opportunities for the chondrichthyans and for the actinopterygians that they were able to take advantage of and really radiate quite dramatically.
Yeah, exactly. It's like you take one piece out of the equation, then you have a lot more opportunities open up for the prey of the placoderms and also for the predators
Yep.
of placoderms, of which there were a few. So
Yep.
really major shifts to the biodiversity and the ecosystem of the Devonian period.
Yeah.
Yeah. Okay, thanks for that. Um, Okay, so continuing on from that last question, so what do you think would have happened if the Hangenberg and Kellwasser events didn't actually happen when they did? So would the oceans be completely different to what they are now, or or something similar?
Well, I'd like to think that instead of just looking at leucorrhagia in the lab, we would be raising placoderms in the lab. So maybe some of them would have survived to the present day. Well, obviously, if the placoderms had continued to live beyond the Devonian, they would be a major, I would assume, a major part of any ocean and freshwater system. So placoderms, as I said, were an extremely successful group. They lasted from the Silurian to the end of the Devonian, and they occupied both marine and freshwater habitats. And they also have been found on every major continent. That includes the Antarctic. So one of my PhD supervisors in Australia had actually done some of the initial work in the Antarctic to find Devonian rocks and then to find
Wow.
placoderms in these Devonian rocks. But otherwise in every other continent. And so they were extremely successful. So I would think that if they hadn't gone extinct, they probably would have continued to be successful, competing with the chondrichthyans, competing with the bony fish. And it's kind of an interesting scenario to think that maybe they would be the dominant animals in these marine and freshwater systems. So in the Cleveland Shale, which is known from Ohio, Devonian age, we have Dunkleosteus, which is one of the largest placoderms that we know about. It's a very huge animal. And there is some thought that one of the other jawed vertebrates that's in the Cleveland Shale is a small shark called Cladostalache. And it's thought that Dunkleosteus might have preyed on the shark Cladostalache. So I would like to think that if the Devonian extinctions hadn't happened, we may still have Dunkleosteus swimming around the seas today, preying on fish. And the whole tone of these nature shows would be very different if we had placoderms.
I would love to see
In the waters
Dunkleosteus in
today.
action. That'd be great.
Wouldn't that be great? Yeah.
It's
So I think
literally like a living can opener. Well, it was.
Yeah, exactly. So who knows? If the extinctions hadn't happened, of course, that's a long time since then till now. Other extinction events, like you said, the Permo-Triassic and Cretaceous, but would be interesting to think that placoderms would still be alive today.
Yeah, I think many people would be quite excited by that. I know I would be, and
I would be
you, of
extremely
course, would
excited.
be. Yeah.
Today, I just look at catfish and think that these are modern analogs, at least, of placoderms in some ways.
Yeah, yeah. And it's weird to think, I was just reading an article the other day about tetrapods, and it's weird to think that snakes are tetrapods. Like, they don't have any arms and legs, but they still are tetrapods. That's pretty
Well,
cool.
that's true, but if you look in fossil record, fossil snakes do have legs.
Well, yeah. As in the extant snakes today, they don't have arms and legs, but their ancestors did. Yeah.
Exactly. Yeah.
Okay, that was quite interesting, actually. So moving on from the Devonian period, now a more general question for you. So with the advancements in technology that we see today, such as CT scans and morphometric analysis being used in paleontology. Do you think this gives a better insight into the past or into the future?
Um, Probably both. I mean, certainly working on fossils, CT scanning has revolutionized the research, and that's not an understatement at all. It's completely overturned the field. So, I mean, you can imagine in the past having to prepare fossils and just, out of the rock, and how time consuming that was. And you had the potential to break fossils as you were preparing it, just human error. But
Yeah.
now you can just take your rock, put it into the CT scanner, and then you get your scan. And if you're lucky, if you've done the scan well, you get a good contrast between the bone of your fossils and the surrounding rock. And if you have that contrast, then you can easily, virtually, using various computer programs, remove the fossil bone by bone from that rock and make a virtual 3D representation of it. And not only CT scans, but also synchrotron scanning used to be, it's a more powerful type of CT scanning. It used to be quite difficult to get synchrotron scans done, but now it's becoming itself more and more common to just put your fossil straight into the synchrotron scanner. And the nice thing about synchrotron scans is it gives you a lot of detail about the structure of bone through the fossil record, for example. So you don't have to necessarily section a fossil anymore. You can just do the synchrotron and get that detail as well. And just also for being able to identify the bones in a difficult fossil, synchrotron scanning helps just so much. So now we have this quite accessible way of studying fossils that we can apply across the board: plants, vertebrates, and with a little bit of work put into what we call segmenting the fossil out of the rock, we can just do so much now with how we study fossils. We can reconstruct them better. We can separate the bones virtually, put them back together virtually. It's just been amazing to be able to do this.
Yeah, there's just a lot of innovation that's come around in the last few decades or so, especially for subjects like paleontology, which is great to hear. I mean, for my EPQ, which for the audience who don't know, and it's basically an A level that you can do, an optional A level, where you write a dissertation or do a presentation on a topic of your choice. And my EPQ is about biomechanics in paleobiology and its uses and whether it's relevant or not. And I had to read a lot of papers about using technology, because biomechanics is predominantly technology nowadays. And I was just really excited by that, because I'm hopefully considering going into paleontology in the future, and being able to use that technology just seems quite exciting.
Well, I know we've talked about looking at Acanthostega, for example, Devonian tetrapod, and other related tetrapods, and how they sort of the transition from water to land. And so to do that work, you're going to first of all have to CT scan Acanthostega, and then segment the toes and the feet and the limb bones out of the rock. So that is just before you even begin to do your your biomechanical study. You'll need to do this CT scanning and segmentation to get the separate bones that you can then subsequently work with. So you might say that fields like biomechanics wouldn't have been as possible, or not as possible to do the range of work that you can do without CT scanning.
Yeah, I mean, they're also interlinked together, so that if you take one bit out, you can't really do the next bit in the process. So that does make quite a lot of sense. But yeah, when it comes to Acanthostega, just the lack of evidence present and just the state of preservation of the fossil just makes it quite difficult to analyze in the field and in a lab. But just
I have to say, the person who did prepare Acanthostega and quite a lot of the other fossils for Jenny Clack who worked for her had done a fantastic, just an absolutely amazing job with the preparation initially. So I don't want to got to remember her contribution as well. But with CT scanning now, you can do a lot of, or you don't have to have as much preparation as you used to.
Yeah, yeah, that the manual element is kind of removed to a small extent, but yeah.
Yeah.
Okay,
Yeah.
so going on to biomechanics a little bit more, so how important do you think biomechanics really is in the grand scheme of things in understanding more about our origins as fish on Earth and also the future of life on Earth in general?
Well, I was thinking about this, and because we are all fish, most of the interesting things in terms of the evolution of vertebrates has actually happened in fish. So if you want to understand things like, you know, how did you get your skeleton? How did you get your brain? How did you your muscles? How do you walk on land? How do you swim? You need to go back to look at fish. And to get a better appreciation of those features, you need to do biomechanics, of course. So again, just to go back to Acanthostega, you're interested in how Acanthostega walked, because Acanthostega, of course, has the limbs, has feet, but it's got more than 5 digits. So how does having those larger number of digits affect the way that Acanthostega swam, or how did it move on the sediment compared to an animal that had 5 digits? And I don't think we get fixed to 5 digits until we get up into the Carboniferous. So
Yeah.
it is a pretty major change. And again, you know, you would need to do the biomechanics of that to find out what the implications of 5 versus more than 5 were during that transition.
Yeah.
So for that kind of question, it's extremely important to be looking at the biomechanics. Of course, you need to have good modern models of movement that you can then apply, because you need to know the parameters to apply to the fossils. So you need to study the living animals as well. But I just also wanted to talk briefly about another project that we're developing, which is looking at the evolution of the neck. Okay. So of course, why, here I'm moving my head around, why is a neck important in the evolution of animals? So if you think about fish in general, their shoulders, where their fins are quite tightly attached to their skull. There's a series of bones which runs from the shoulder up to the skull. And it's in Tiktaalik, actually, okay. Is one of the first fish. There are some other examples, but it's one of the first closely related fish to tetrapods, which shows that that connection has been lost. So Tiktaalik can move its head with respect to its fins and its shoulder. So suddenly you have the ability to move your head around, even though you're still in water, to move your head around to see predators. And once you get onto land, that has huge advantages, of course, to be able to do that. Yeah. But to go back to placoderms, we're interested in looking at neck movement in placoderms. So in placoderms, you have the head is covered in bony plate, as is the front part of the body, which we call the trunk shield. Now, the head can actually move on the trunk shield at a joint, so it can move up and down like this. Yeah. Of course, those sorts of movements are important in things like feeding, because if you can raise your head shield, you can drop your jaw, and in that way, you can greatly increase the size of your mouth, your oral cavity. If you can do that, make your mouth bigger, you can eat bigger prey items. So that kind of movement is really important. And of course, placoderms are one of the first, phylogenetically basal or primitive jawed vertebrates. Yes. So again, this is occurring when we first see jaws, when we first see paired fins, we have a movable neck. But if you look carefully at that joint in the variety of placoderm groups, they're quite different. So different in whether you have a knob on the trunk shield that meets a concave surface on the head shield, or vice versa, different kinds of joints. And what our project was interested in is just knowing whether that difference in morphology among placoderms with the neck joint had something to do with the function of the neck joint. So this is where you need the biomechanical study to look at how in a long extinct animal that neck was functioning. So again, that's a project that we're developing at the moment, but that could really say something about, you know, in these early jawed vertebrates, were they already showing quite different morphologies, quite different functions in feeding, different environmental niches. And that's where the biomechanics comes in. That's very important.
Yeah, that makes quite a lot of sense actually. I wasn't aware of that project, but that does sound quite interesting indeed. Especially just finding out, because we take these things for granted, I mean, of course, but finding out where they came from, how they came about, answering those
Yeah.
key questions, and
Yeah.
what the point is in having specific organs or bones. I think that's very important to understand.
Well, it's easy enough to say, well, there's a joint there, it must move up and down, but it comes down to biomechanics to look into that more deeply and appreciate it. You know, if there's a range of motion, are there important differences? That's where the interesting information comes from.
Yes, exactly. And then you can make more calculations about that later on, about its diet and also a niche in its environment.
Yep.
But yeah,
Yep,
that's a
indeed.
great thing about the placoderms. They were so wide ranging and the different
Very,
diets, different niches, everything, which is
yeah,
great.
yeah. And of course, then you can build that together as a data set with their different jaws, different dentitions, different extent of the trunk shield on the body, the types of fins they had. And you can build a more complete picture of how the placoderms lived.
Yeah, that's really cool in my opinion.
Yeah,
Quite
yeah, me too.
interesting. All right, so that is the end of the questions that I had prepared for you. But is there anything that you'd like to say to the audience? Maybe something innovative that you think they should know, or just anything at all?
Well, I thank you for again inviting me to this interview and for
Thank you.
these really very interesting questions, because it made me think and sort of made me probe into different areas. But I just would like to say, you know, that I think the study of early vertebrates is so important, and I've been really lucky to be able to do this for a job at a great place like the Natural History Museum, because not only do we have all these great research facilities available, but also extensive collections here to work on. So we have one of the top fish collections in the world. And so, you know, I would encourage you that if you do have an interest in vertebrate evolution, that you don't, please don't, no, after Mike Benton was here, please don't study dinosaurs, come study fish. No, you might want to edit that one out later, I
No, no, I'm going to keep that in there just so they know. Yeah.
think. No, I shouldn't say that, but I mean, studying early vertebrates, as I said, that's where many of the major evolutionary changes that really impact upon us today have occurred.
Yeah. Well, thank you, Dr. Johanson, for your time. Thank you for coming here today. Those were some really great responses from you, and I hope that the audience have learned something new today, whether they're in paleontology or not, or a different field. Hopefully, they're encouraged to look more into your line of work and the similar fields and interests as well. So, yeah, audience
Okay,
members, stay tuned for the next episode, hopefully coming soon. Um, and yeah, so once again, thank you, Dr. Johanson. And
my pleasure.
yeah, thank you very much.